Some MCQs of cell-mediated immunity. Which of the following is function of CMI? Immunological surveillance, delayed hypersensitivity, killing intercellular parasites, or all of the above. The cell-mediated immunity participates in immunological surveillance. It is also responsible for delayed hypersensitivity, and it participates in killing of intercellular parasites. So the correct option here is option D, all of the above. Which of the following cells has least association with CMI? T cells, large granular lymphocytes, B cells, and monocytes. So T cells, monocytes, and large granular lymphocytes are actively involved in cell-mediated immunity, while the B cells, they participate in humoral immunity. So the correct option here is option C, B cells. All of the following are properties of T cell independent antigens except antibody response is IgM or IgG3. They are slowly metabolized in the body. They create immunological memory, or they are structurally simple. T cell independent antigens, they fail to produce immunological memory. So the correct option here is option C, create immunological memory. Question 4. All of the following are examples of T cell independent antigens except erythrocytes, pneumococcal capsular polysaccharides, bacterial lipopolysaccharides, and flagellar protein flagellin. Option B, C, and D, that is pneumococcal capsular polysaccharide, bacterial lipopolysaccharides, and flagellar protein flagellin are T cell independent antigens, while erythrocytes are T cell dependent antigens. So the correct option here is A, erythrocytes. Which word best describes peptide mediators or intercellular messengers which regulate immunological, inflammatory, and reparative host responses? Option A, monokines, B, lymphokines, C, cytokines, and D, interleukins. So monokines, lymphokines, interleukins, these all belong to a big group which is known as cytokines, which describes best these kind of mediators. So the correct option here is option C, cytokines. Which of the following is a function of interleukin 1? Stimulation of T cells, B cell proliferation, chemotaxis, and all of the above. Interleukin 1 stimulates T cells and B cells both, and it is also participating in chemotaxis. So the correct option here is option D, all of the above. Which receptors are present on the surface of T cells? T cell receptor, microvilli, surface immunoglobulins, or all of the above. So surface immunoglobulins and microvilli are present on the surface of B cells, and T cell receptors are present on the surface of T cells. So the correct option here is option A, T cell receptor. CD4 T cells are divided into following subsets: A, Th0, Th1, Th10, Th11, B, Th0, Th1, Th2, Th3, C, T41, T42, T43, Tr4, D, Th0, Th2, Th4, and Th6. The correct option here is option B, Th0, Th1, Th2, and Th3. Following is true about natural killer cells and lymphokine activated killer cells. They are not B cells. They kill fresh tumor cells. They do not require recognition by MHC, and all of the above. So natural killer cells are not B cells. They kill fresh tumor cells. They do not require MHC recognition. And so the correct option here is option D, all of the above. T suppressor cells are known as CD4 plus CD8 minus T cells, CD4 plus CD8 plus T cells, CD4 minus CD8 plus T cells, and CD4 minus CD8 minus T cells. T suppressor cells are CD4 T cells. So the correct option here is A, CD4 plus CD8 minus T cells. And this is the answer key. Thank you.